Welcome to the presentation of my poster with the number B004. My name is Florian Gisdon and I'm a postdoc in the molecular bioinformatics group of Professor Ina Koch at the Goethe University Frankfurt. I am working on the graph theoretical analysis of proteins, and I will present our approach to predict functional biological modules in large protein complexes. The approach was mainly conceived and developed by Marilla Sunka and Jan Niklas Wolf, while I continued the development and the analysis under the supervision of Jörg Ackermann and Ina Koch. On the poster you can find an illustration of our approach, which we applied in 3 different case studies. The functional complexity of biochemical processes is highly related to the interplay of proteins and their assembly into diverse protein complexes, as shown here for complex 1. Proteins first assemble into functional units, which further assemble into a multifunctional protein complex, which then functions within a biochemical process, here the respiratory chain. In recent years, the discovery and characterization of protein complexes has substantially progressed by advances in cryo-electron microscopy, proteomics, and computational structure prediction. It is essential to have approaches to process this number of data and to extract relevant information for structural and functional characterization. Our hypothesis was that functional modules within protein complexes can be predicted based on the context between protein chains. As basis for our approach, we modeled large protein complexes as weighted complex graphs using the PTGL tools, a software which was developed in our group. Here you see a screenshot of the PTGL web server, which can be used to explore and analyze the graph structure of many proteins, and you also find the link to the GitHub repository for the PTGL tools. We first computed the complex graph with PTGL tools, and then we applied the Leiden algorithm as a graph theoretical clustering approach. The basic steps of the Leiden algorithm are the movement of nodes between the communities and a refinement. Starting from a singleton partition in A, B shows the initially computed partition and C the refined one. Based on the refined partition, an aggregate network is created, shown in D. However, the communities in the aggregate network are assigned according to the initial partition from B. For example, the red community in B is refined into 2 communities in C, and after aggregation, they define 2 nodes which belong to the same community. This procedure of movement, refinement, and aggregation is repeated until the best partition is found. For the analysis, we are starting from a quaternary structure in the MMSIF format. Using PTGL tools, we are computing the complex graph, which has vertices representing protein chains and edges representing spatial contacts between the chains. The edge weights, which are shown here in the graph with a line width and a line color, are representing the number of contacts between the chains. With that representation of the edge weights, well-connected regions can already be observed. We then applied the Leiden algorithm as a clustering approach to compute a partition of the complex graph. According to our hypothesis, the partition of the complex graph should match the functional biological modules in the protein complex. The partition is then mapped onto the quaternary structure of the protein complex. Starting from the quaternary structure of a protein complex, our results comprise the complex graph, the computed partitioning, and the structurally assigned modules. We applied our approach in 3 different case studies and considered different aspects for the analysis. In the first case study, we investigated the complex 1, which is the first protein complex of the respiratory chain. It has 45 protein chains, of which 44 are unique. The functional biological modules N, Q, P proximal, and P distal have been assigned experimentally. The well-connected regions in the complex graph match the biological modules. The computed modules M1, red, and M2, green, from our approach correspond to the N and the Q module, respectively. The computed module M4, yellow, represents the distal P module, which harbors 2 of the 3 antiportal-like subunits for proton translocation across the membrane. The computed modules M3, blue, and M5, purple, together represent the proximal P module. The module M5 is associated with the 3rd antiportal-like subunit, while the module M3 might correspond to an assumed 4th translocation pathway. In the second case study, we analyzed mechanistic and dynamic aspects of the chaperonine trig CCT, which is mainly associated with the folding process of actin and tubulin. Trig CCT has 2 rings, and each one consists of 8 parallel chains. It is a highly asymmetrical protein complex despite its pseudosymmetrical appearance, and its mechanism relies on the asymmetry. We analyzed 2 conformations which appear in the mechanistic cycle. Both conformations show an asymmetry in the graph structure, which correlates with the experimentally assigned hemispheres. We additionally identified a key subunit of the complex, which also changes its affiliation to the computed modules. In the third case study, we investigated the hat of the bacteriophage Phi29, which is a very large protein complex with 400 protein chains built out of only 2 unique chains. The structural elements of the capsid are hexameric and pentameric capsomers, which are formed by one of the 2 unique protein chains, and trimeric fibers, which are formed by the second unique protein chain. The structural elements are visible in the graph structure. Additionally, the predicted modules match with about half of the asymmetric unit and might be interpreted as pattern. We introduced an approach to predict functional biological modules in large protein complexes based on the context between the protein chains. Using a quaternary structure as input, we computed the corresponding complex graph, which showed well-connected regions or even central structural elements. And the graph theoretical partitioning of the protein complex graph correlated with functional biological modules. In conclusion, our approach can be used to predict biological modules of protein complexes and might also be used for their classification. Thank you for your attention.